So I want to introduce the speaker of today, and I need to start by welcoming you all, and thank you for choosing to connect to this webinar presentation. This is Genomic Tools for Sweet Potato project, in which we are organizing a series of webinars. This is the first one to inform and educate the sweet potato breeders in Sub-Saharan Africa and global in the world, and also to educate other communities of practice who are also working on crops or plants which have complex genetics like the sweet potato. Our speaker for today will be Craig Yencho, who is a distinguished professor and a program leader sweet potato and potato breeding and genetics program in North Carolina State University, and is also lead PI of this project, the Genomic Tools for Sweet Potato project, which is funded by Bill and Gates Foundation. Our assistant speaker will be Dr. Mark Gishling, and he's joining us from wherever he is. And Mark Gishling, he's the Genomics and Biotechnology Global Program Leader, and he's based here at SIP Nairobi. His research focus covers the use and characterization of potato and sweet potato, wild varieties, genetic mapping of traits of interest, isolation of genes and alleles, among others. So welcome all. That you type your question, and we'll leave it at here Nairobi. We have 30 minutes for Craig to do the presentation, and then the 20 minutes or 25 minutes, 20 minutes for addressing the questions. So thank you and welcome all. Thanks, Craig. Back to you.
Thank you very much, Mark, for the action. And good morning, everyone. From North Carolina, it's a beautiful, crystal clear spring day here. And I hope that wherever you're tuning in from, that it was a good day there. I realize that some of you are ending your day. I'll jump right into things. I don't do webinars very often, so this is a bit of a learning curve for me. And if you have questions or if I need to slow up, I'll assume somebody will type a comment to me and send it to me. I'll be able to see it on my screen. So my title for today is an overview of the what we call the GD4SP project. Overall goals are improved breeding technologies and increased genetic gains for sweet potato. If I need to get yep. I'll try to go slow. I'm continually impressed with the range of diversity that we see in sweet potato and the range of crops create from this remarkable crop. We're seeing a lot of feedback from somebody. If people could mute, I won't have the echo then coming in on my phone. Of course, we all know sweet potatoes are a very important food crop globally, and this is just a picture that I pulled from FAO recently showing the global distribution of sweet potato and the major production countries. And of course, what you see is that China is a major producer of sweet potatoes, and the Southeast Asia region. And sweet potatoes are important all across Africa here, but it really is not so apparent from this figure. And then, of course, sweet potatoes in North America are actually fairly minor crops. But I happen to live in a state where they're very, very, very important for our economy. I came from the International Potato Center several years ago, and it shows area cultivation and average yield of sweet potato across the globe. And what you notice here is, in terms of yield, average yields in, say, China, the major production regions, are actually quite high compared to the average yield across sub-Saharan Africa for sweet potato. And our goal is to try and change this situation right here. And I do believe that if we are able to change this, then you're going to see that sub-Saharan Africa is actually going to be the major producer of sweet potatoes across the globe. And I think we can do that in less than 10 years' time. The frame of reference for my audience is generally as to what my situation is like here in North Carolina and the United States with reference to sweet potato. And in fact, we have quite a fascinating story to tell. I was working with sweet potato back in 1997, so it's a little bit off the graph. In that short period of time that I've been working in sweet potato, we have seen the first harvest of a sweet potato increase twofold. We've seen total production of sweet potato during that same period of time increase roughly threefold. And we've seen that yields, average yields of sweet potato across the United States, and particularly in North Carolina, gone up significantly during that course of time also. Now, I think many of you know I'm a sweet potato breeder, and the focus of my program is largely developing new varieties. And we've been very fortunate. In 2005, we released a very popular variety called Covington. It probably occupies roughly 40% of the total U.S. crop right now, and probably about 80 to 90% of North Carolina's crop. You can see that upon releasing Covington's actually benefited through this tremendous growth in our industry during the course of the year. I would love to say that Covington was the driver of this growth, but simply not true. Actually, the driver of growth in the U.S. market has really been two factors, actually three factors. One, we can store the crop all year round, and that is we can provide a continuous supply of sweet potatoes to various market sectors at any time of the year. And this has been a real enabler in this tremendous growth that we've observed. Now, here I'm showing you just a kind of a sampling of the range of sweet potato products that are currently being made from sweet potato. But truth be told, the main drivers of sweet potato product expansion right now are actually the production of sweet potato French fries. In Africa, we would call them chips, and also sweet potato chips. We would call them, you might call them crisps in the UK and Africa also. So these two products have been huge drivers of increase in sweet potato production, the demand for sweet potatoes, because these two market sectors have really been growing, but also producing juices, other novel food products, even dog chews, and even beers out of sweet potato, and also some distilled products. We even make a vial of sweet potato nowadays. So this is the reason why our acreage has increased and our per capita consumption has increased. Also, we're the same thing in Africa too. The crops are growing in importance, and there are also a lot of new value-added products that are being developed from the sweet potato across the continent. And this is just one example that Karimi sent with me, shared with me at the at a meeting that we had in Rwanda just last fall. These would be these golden power biscuits and fried donuts, or mandazi, made from sweet potato puree. So if you recall the same graphic that I showed you for the U.S., these are the same production trends. These are the production trends for sub-Saharan Africa. And what I point out to you is there's a lot of good things happening in sweet potato across the continent also. One, the harvested hectares of sweet potato is increasing. Two, production is increasing also. But something that's really different compared to our more developed system here in the U.S. is this yield line right here. Essentially, since the early 1990s, yield trends across sub-Saharan Africa have been fairly flat. If we're going to improve production in sub-Saharan Africa and continue to increase the profile of the crop, we need to bend this production curve upward and increase per acre yield. And partly that's the whole reason for the development of the genomic tools for sweet potato improvement project. So our goal is to change this equation, change these brown and red marks into yellows and greens, and thereby improving total production and creating great other opportunities for sweet potatoes across the continent. There's good that has been done with sweet potato their last decade, and maybe decade and a half or so. One project I'd like to mention is called the SASHA project, or it's called the Sweet Potato Action for Security and Health. It's actually funded by the Bill and Melinda Gates Foundation. I was very fortunate to be an advisor to the foundation on this project in a technical capacity. This project was led by the International Potato Center, and if you're seeing the picture on the screen right now, I'm sure you'll recognize a number of the players there. It's made major progress in 4 key areas. One, in developing improved populations and in developing new varieties. Making good progress in seed systems. Delivery systems have been explored, some proof of concept market delivery systems. And the SASHA project really has been very successful in developing a series of sweet potato support platforms around the continent of sub-Saharan Africa in Uganda, Mozambique, and Ghana. And program leaders there would be the lead for the Uganda program would be Dr. Robert Mwanga right here, Dr. Maria Andrade down in Mozambique, and Dr. Ted Carey over in Kumasi in Ghana, and of course Yencho as overall project leader based in Kenya. I'm not sure who's all attending the webinar today, but if you're here and I just mentioned your name, I know Robert is here. A quick shout out to you guys. You've done great work and continue to do so in the future, I'm sure. There's another initiative that has developed fairly recently. It's several years old now. It's called the Sweet Potato for Profit and Health Initiative. The FPHI is a multi-partner, multi-donor initiative that seeks to reduce child undernutrition and improve smallholder incomes in 10 million families by 2020. Folks, it's 2016. So we've got a big job ahead of us if we're going to reach this goal by 2020. And I can tell you that I think we're well on our way to that target at this point, and perhaps that can be the future webinar by one of our team members to describe that. Now, the agency for SASHA and the FPHI is the International Potato Center, based in Lima. Largely, a lot of these efforts are led out of the substations in Dhaka or at ILRI in Nairobi, and of course led by Jan. NC State is a member of the Sweet Potato for Profit and Health Initiative, and with the support of the foundation, we have been able to develop the GT4SP project, and I'll tell you a little bit more about that in just a few minutes. Let's sit back for a moment and take what I'd call a sweet potato global status check. I think it's fair to say that the importance and potential of sweet potatoes is becoming widely recognized across the globe. Next week, I'll leave for a trip to China to discuss sweet potato, and literally, I have traveled around the globe several times to discuss sweet potato, and it really is an up-and-coming crop in many countries. So many public and private organizations recognize the superior nutritional value in sweet potato compared to many of their staple crops, and investments are really increasing. SASHA 1, as I mentioned before, has been extremely successful. It was renewed and funded. SASHA 2 and the Sweet Potato for Profit and Health are gaining more traction all the time and gaining new partners all the time. And if you're not familiar with the Sweet Potato Knowledge Portal, I would reference you to that portal to learn more about all things sweet potato across sub-Saharan Africa and indeed the globe at this point. It's a great website for sweet potato. So in short, a lot of people are more interested in sweet potato than before. Reader. And when I survey the situation and the genomic resources for improving sweet potato, they're noticeably lacking. And that's the focus of the GT4SP project, is to improve genomic resources available for sweet potato. Now, let's just take a quick look at what's out there right now, and this is certainly not an exhaustive look, but it's just a quick and dirty look at some things that we've done here at NC State in collaboration with the International Potato Center over the years and some national agricultural research partners, namely the National Agricultural Research Organization in Uganda and at Nakri. But first, let's do a quick Google search, which a colleague of mine, Awais Khan at SIP, has done recently. Just a quick Google search on various crop search terms, for example, crop and genomic, or crop and genomic for 2014. You're going to see is compared to the other major staple crops, wheat, rice, soybean, sugarcane. You're going to see just in terms of the number of hits, when you do a quick search on Google Scholar, is really low compared to many of the other major crops. You compare it to cassava and yam, it's fairly low. So we have a lot of work to do. Now, to give some credit to some of the early funders of the sweet potato genomics work, and that goes to the McKnight Foundation Collaborative Crop Research Program. Actually, my program, the University of Greenwich and the National Resources Institute, and Namalangi, or the National Crops Resources Research Institute in Namalangi, Uganda, have been engaged in a long-term capacity development and breeding project about the past 8, well actually I should update this, it's not the 8 to 10 years, we've been going back all the way to 1996, so we've had a long-term relationship for 20 years now, and the focus of the effort has been to develop high-yielding, high dry matter, orange flesh cultivars with increased resistance to viruses and insects. And really the guy who got the ball started all of this is Dr. Robert Mwanga. And just last week I had the great pleasure of welcoming Robert back to our institution here at NC State, where we graduated after 15 years of being a, and I think he'd say that campus has changed remarkably, but he really got a great project started, and it persists until today. So I want to give Robert a lot of credit for that. Our focus, like I said before, was development of high-yielding multiple resistant sweet potato germplasm. We focused on the orange flesh high dry matter type, and across sub-Saharan Africa, the viruses, virus disease, and sweet potato weevil continue to be real scourges, and they're actually going to be a major emphasis of the GT4SP project. These are still major biotic constraints to sweet potatoes in Africa. This would be anthracnose in sweet potato also, primarily a problem in the highlands of Africa, another issue that we'll be wrestling with, but really these two up here, the viruses and the weevils, if we could do something to change this and develop cultivars with improved resistance to those two pests, it would be a major step forward. So a little bit of working in genetic mapping. Two people that have led that effort here would be Dr. Jim Carlos Cervantes, who's now a ferret breeder with Vilmorin, and Dr. Bernard Yada, who's now back at Nakri and leading the Ugandan sweet potato breeding program. And here's a picture of Robert some years ago in one of his polycross breeding nurseries. And then Dr. Gareta Semakula has picked up and is working in the project also. We were through this project to complete the first complete genetic linkage map of sweet potato in a population tanned by Beauregard. And this was a great first step using amplified fragment length polymorphism. And in that work, we were able to tag QTL for various traits. And this is one example of QTLs for, this is dry matter on the red bar, this is starch on the green bar, beta carotene is up here, and what we're able to confirm is a negative linkage between dry matter and beta carotene. In bars here with an underline on them, that means the QTL is for reduced dry matter and reduced starch. This is a locus for increased beta carotene. And all the applied breeders who've been working with sweet potato over the years realize that there is this negative linkage. It's hard to get high dry matter, high orange flesh variety. I believe it's fair to say we've now broken this linkage, but certainly it hasn't been due to the use of molecular markers. It's been to all applied genetics and lots of creative and large-scale breeding efforts, large by our African breeders. But nevertheless, we still are not able to use molecular markers in sweet potato breeding, so we need to, and this is what our project is focused on. So we've concept, we can make genetic maps in a complex hexaploid polyploid. We can tag QTL. The next step is to begin to use those to facilitate breeding. Recognizing this, the foundation realized the opportunity that existed. We had a convening at the Gates Foundation in Seattle back in June of 2013, and we understood that sweet potato is poised for significant growth in sub-Saharan Africa. But in order to fully realize the long-term potential, the foundation recognized that we needed to invest in modern breeding tools, integrate these modern breeding tools into applied breeding efforts that are connected to fruit seed systems and market-based value chains. And so what I would say is that the GT4SP project is very tightly connected with ongoing investments by the foundation in the Sasha project and the seed system project they started, and many other projects that are ongoing. We're well connected in that effort. So out of that meeting, the GT4SP project was born. It's a large project. As Mercy mentioned before, I am the PI, and it certainly changed my life. In terms of the scope of activities, we encompass over 20 PIs, 7 institutions, 6 countries, 4 continents, 15 time zones, 1 crop, and a $12.4 million investment. All of this is for breeding tools for sweet potato. What's our vision for the project? Our vision for marker-assisted breeding in sub-Saharan Africa includes the following. 1, in order to develop robust genomic tools for sweet potato, we need good genomic resources. That means we need a reference genome. We need new, robust SNP markers. We need advanced laboratories capable of sequencing, developing, and linked with developed and developing countries so that we can work together as a team. We need both 2X, cultivate or 6X mapping populations, and we need training and test populations. With that, we need to develop new phenotyping, analytics, and database resources. Look at this as a pyramid. Certainly the bottom of our pyramid, and probably the most important part of it, is we need human resources and we need capacity development. And we have a lot of that built into the GT4SP project. The Sasha project and other projects have the similar built in. And that's partly what this webinar is intended to do. We need to continue to assemble and develop a dynamic team of breeders and allied disciplines to help us. We need training in the use of traditional and genomic breeding methods. We need to work on our communication and our collaboration. We need institutional training and capacity development. And largely this institutional training and capacity development is being led by Mercy and her team, Dr. Mark Gilon and Mercy at Becca at the SIP station in Nairobi. So here's a cartoon, if you will, on the various aspects of the GT4SP project. we have a team that's working on establishing a sweet potato genome. I'll give you a reference genome for sweet potato. I'll give you some information on that. We have a bioinformatics group that is focused on developing the bioinformatics and analytic tools that enable us to sort of decipher the reference genome. And we have a genotyping by sequence platform that's being initially set up here at NC State. Our long-term vision is probably that's going to be trend to Becca in the long term through some other investments. A database, bioinformatics and phenotyping team that's set up that is focused on receiving all of this data, assembling it together, and making it useful for breeders. And then have breeding and capacity development efforts that feed back into all of these higher level components. I currently have 2 PhD students with me right now, one from Uganda and one from Ghana. They're working, started up in August, and they're working on mapping populations that are being developed on the GT4SP project. And they're both coming from national programs, the CRI in Ghana and NARO in Uganda, and the intent is once they finish their research here, they'll go back to their field work at their respective locations, come here, complete their PhDs, and then return to their home countries and continue to work on sweet potato. So, ambitious project, there are a lot of pieces to it, but so far it's going pretty well. And give you a couple of updates on it. We had our project meeting. It was our start meeting back in San Diego in January 2015. You can see this is the core group right here. Just this last January, we had our first annual meeting, and you can see that the group has grown in strength and in numbers. We've brought together a really great team of people to bear upon sweet potato, and I'm really pleased to be working with this whole team. It's truly an honor. We had our first joint SACA/GT4SP joining in Mukono, Uganda. That was last year in June, and we're getting geared up for our second joint meeting, which will be in Nairobi this coming June also. Many of you, I think, know about it already. I want to Ted Carey a quick shout out here if he's on the call. Ted brought along a bunch of orange sunglasses, which we all agreed to put on, and I think it's just it's kind of a fun moment for us to realize we're all looking through the same common lens, if you will. Orange sweet potatoes to improve vitamin and nutritional status in across the continent, particularly in women and children. So I got a good laugh out of this one. We all came home with a pair of new lenses to look through. A couple of highlights for you. You know, as a project leader, when you manage a project this big, just getting all the subcontracts worked out and sorted out is a major milestone, so I had to put that out. All of our subs are all done and negotiated, and all the funds have been allocated. We actually did our first annual report back in September. During the report, a total of 60 milestones in whole results framework. 13 milestones were completed and achieved in year one. And we're off to a really good track. We've hired a project coordinator. We have 8 new PhD scientists hired, 4 PhD graduate students, and greater than 15 support staff. At the meetings that I've mentioned before, we have 2 new websites, a Sweet Potato Base site, which is our database website for the breeding database information established at the Boyce Thompson Institute. And project website, which is at sweetpotatogenomics.cal.ncsu.edu. Substantial progress on establishing a reference genome for sweet potato. I'll go over that in a couple seconds. We first generation genome browser that has been released, and it will probably be publicly available within the next, maybe next month to 90 days. Both diploid populations that are established between a wild relative of sweet potato, Iphitrifida. We have started work to make a map out of these 2 populations, and what I can say is actually that map is completed at this point, but we're in the the process of tweezing it. We have a lot of great cultivated populations that have been developed, and we have another population which I'll call the Mwanga Diversity Panel, consisting of a set of 16 sweet potato parents crossed in a dial that we'll be using for doing some genomic selection work in. This is going to be a great set of materials in the future as we move forward, and that's being established. All of these populations are actually being established in tissue culture, so it can be shipped across national borders. And of course, our capacity building efforts have begun now at Becca. Quick look at the materials that we chose to do our reference genome from. There are two putative ancestors of cultivated Iphypetatus, cultivated sweet potato, Ipomoea triloba and Ipomoea trifida. This is the triloba line, and this is trifida line, and these are the M-curve plots. And basically what these plots show you is if they have single high peak, they're highly homozygous, and they'll be suitable for sequencing and then reassembly, just because it's a little bit easier to reassemble a very homozygous line. And this triloba is particularly good for that purpose. The trifida is not as good as the triloba, but it's suitable. And interesting that both of these lines came out of our breeding program here at NC State, a totally different project, our ornamental sweet potato breeding program. These will be the reference genomes for sweet potato. The genome assembly results done by Dr. Zhengjun Fei at Boyce Thompson Institute. And what I'd like to point out to you is that they're good, they're high quality genomes covering about 82 to, well, 8%, 90+% of the genomes of each one of these diploid species. The estimated genome size are down here, about 526 megabase for Ipomoea trifida, and about 495, we just call it 500 for I triloba, so roughly about the same. This is all Illumina-based sequencing. Packed bio long reads to reduce the Illumina-based gaps, and what we found is that in this genome here, the triloba, which was actually quite good, the Illumina-based gapping, didn't really make much more of a difference in terms of the assembly. It helped, of course, compared to the trifida here, where the assembly was improved significantly more packed bio approach after the Illumina sequencing. So we closed those gaps with the long packed bio reads and basically have improved our assembly. This summary of the assembly provided by Dr. Fei. I won't details here, but I would like to say is that it looks like both the trifida and the triloba are suitable for use as a reference genome for the cultivated sweet potato. And it looked like that cultivated sweet potato probably consisted of two ancestral genomes, one a tetraploid trifida, and second a triloba. So this would be the predominant contributor to cultivated sweet potato, and triloba lesser so, but we're still exploring the hypothesis, and this would be first out of this project. Dr. Fei sent all of the information to Dr. Robin Buell, who is now putting together a genome browser. We have an estimated number of genes of roughly about 34,000 genes in both species. This is the average gene length and some of the metrics of the genome. We are beginning to functionally annotate those genomes using various strategies. And we developed a nomenclature for each of the locuses. And this is implemented in a JBrowse genome browser. This would be the first genome browser for sweet potato. Again, another first for genomic references for sweet potato. We will be doing training this in due truth be told, as an applied breeder, this to me is a bit braille, but basically it's a great roadmap to identify genes, use those identified genes as potential candidate markers, candidate genes in mapping populations. So it is a very good resource as we're moving forward for sweet potato to understand the complexity of sweet potato, understand the genetic makeup, where genes are located, how they're structured, et cetera, that are very useful moving forward. And we'll be through probably webinars doing some advanced training on how an applied breeder might begin to look at and utilize a genome browser in their era of genomic breeding. I mentioned earlier that at NC State we're developing a genotyping by sequencing platform. That's coming along quite well. Our strategy is to develop both low and high density genome-wide SNP pipelines. Will be at multiple plex, and we assume that we're going to have uniform coverage of silent alleles across the entire genome. See that there are going to be some areas we're going to have high coverage so that we can call dosage in complex sweet potato polyploid. In certain cases, we might not use imputation, but in any cases, since we have a reference genome, we may be well trying to imputate cost based on our knowledge of the reference genome. All right, and we first need to determine that, and we're well on the way to determining that what's going to be the best reference genome for calling SNPs in hexaploid sweet potato. A little bit more strategy. We want to develop both low density SNP maps and high density maps for different breeding purposes, and we're going to make sure that these are reasonably cost effective. They'll exclude repeat reasons, exclude unusable SNPs, and we'll use methylation sensitive enzymes similar to the Cornell protocol, except we're doing a few things in terms of size and in optimizing our enzymes for sweet potatoes. This is a flow diagram of that. I won't go into the details of it right here, but it is the standard GBS protocol for sweet potato. The only thing that we're really changing is we're changing the way we're developing our barcodes. We're using different double digest enzymes, changing barcodes a little bit. We're size selecting, and then we're doing the sequencing, and we're working on that right here. We had a team here last week. It's managing the phenotypic data with the genotypic data led by Dr. Lucas Mueller's group at Boyce Thompson Institute. This effort is now coming along right now, and we'll be instituting some training programs in that. Again, I think a webinar, a web methodology is a very good methodology to do this, but we're also going to be doing a lot of face-to-face training at the individual breeding programs as time progresses. This is just a quick look at sweet potato base right now. It has been constructed. We have yet populated it with much data, and that's going to be the next step, is to really begin to populate this database with mapping experiments that we're engaged in, and have the breeding programs begin to use the database as a tool in their breeding program. This is a real departure from the past, and so there's going to be a lot of training that's going to be required for that. It's a picture of the team that was here last week. It's the database group right here, sitting in front of what we call the Tally Center at NC State. We took a day off to tour the field. Sometimes it's good that we had a mixture of actual breeders in the group, then bioinformaticists in the group. This is Lucas Mueller right over here, leads the database group. Geneticist Awais Khan, Bernard, Robert Wonga back, they're alumni, genomicists. So we took the time to get them out in the field to see how we do it in North Carolina. And for those of you that are unfamiliar with our planting process in North Carolina, these are actually sweet potatoes right here that are laid out in a row in a process called bedding. This cart actually is delivering the sweet potatoes, laying them out on the row right now. These are covered with soil. In the next, these will be covered with soil. The next step in this planting operation will be covered with soil, and then sprouts will emerge from these roots right here. And then in North Carolina, what we do is we cut the sprouts that emerge from the roots and transfer those into the commercial fields. It's very different from what we do in sub-Saharan Africa. I'm going to wind down here And here's a couple of things that I've learned from lessons that I've learned from year one. I've learned how critical it is for a reference genome. And once you have a reference genome for sweet potato, how important it will be. I understand we need a sequence-based genotyping platform. What that will ultimately look like is yet to be determined, but we're putting the flexibility into our project so that we can understand that. We need reader-friendly bioinformatics and analytical environments. And when I say something like this is very easier said than done. What it will look like, what are the special needs for clonally propagated polycrops, and how that's going to look. And I also know that we're going to need technical support for breeders. This is a big change for us in here, and this is going to require a lot of training and a lot of hand-holding. We also have good populations to work with. I'm not really worried about that because we have some great breeders that are engaged with the program, and we're going to be able to develop as many populations as we need. What's really going to be the trick is to incorporate these aspects into our breeding work and incorporate the genome aspects into the breeding work too. If all of this and you're doing more mapping, we need improved phenotyping and data collection capabilities. We all need mechanization. We simply can't handle the number of prodigies by hand anymore. We need sustained funding. And sustained funding is going to have to come from multiple sources, not just one entity. So we as a community are going to have to work on that together. And I dare say that it's not only going to be the sweet potato community that works together to do this. It's going to be the sweet potatoes, the yam, the cassava, all of those other communities working together. We need sustained capacity development, and this is critical. What I'd say is just leave you with those concluding thoughts. And I would say is our project is a very big project. This is a list of the institutions and the lead PIs for each group. It's a great team, and a real honor to work with each and every one of them. And as an applied breeder, really that I can work with such a diverse range of people, and I think it's emblematic of what breeders do nowadays. Breeders don't only work with breeders now. You work with bioinformaticists, you work with programmers, you work with food scientists, you work with pathologists, you work with entomologists. It really is an exciting discipline, and it's a fun job, and I feel fortunate to be engaged in it. So I'll leave, and that's my last slide, I believe. And if I have time, what I would say is stay tuned and be engaged. And I hope that this seminar has helped you to get a slightly idea of what the GT4SP project is all about, and certainly what sweet potato is all about across the globe. Thank you. Maria, you.
Tool is going to develop. Then I'll take your questions. From the list of questions to send on the chat, I have one question. And I think I should open the floor for the questions. And then, because I haven't gotten your questions on the chat, I have to ask from where you are. We still have about 7 minutes to go. And our webinar. And Mark and Craig will answer our question.
Okay.
Craig, yeah, when you are presenting, you mentioned quite a number of traits that the project is focusing on. And from a breeder's perspective, my question is, are you going to focus on the improvement of all these traits traits. And you mentioned high dry matter, or high yield, resistant to diseases and pests. And if you're going to focus on all of these traits simultaneously, what advantage do we have with the breeding tools that the project is developing? That is the reference genome and the mapping function. What advantage do we have if we have the reference genome and the mapping population? Thank you.
The short answer is we are going to focus on all those traits, breeders, but I don't think that we'll use genomic tools to facilitate breeding for all those traits. And perhaps my first two major targets for a reasonable marker-assisted breeding platform in sweet potato would probably be sweet potato virus disease and perhaps sweet potato weevil, if we can identify those that are associated with those two traits. And my reason for saying that is because such difficult traits to breed for already. And if we had a system, a marker or markers that could help us identify progeny early on in the breeding process and advance those, that would be much more beneficial to us. I think we have to be prudent in trying to determine what we can and what we can't do. It's yet to be determined right now how the reference genome will help us, but there's plenty of opportunities for known genes, for understanding if we have a QTL by looking for closely located SNPs or SNPs that are in candidate genes, that we can understand the physiological or physiochemical basis of a trait. So that's where the reference genome is going to come in handy for something like that. In the long term, this emerging concept called genomic selection, where you can identify the value of a particular clone or set of clones and use those to make your optimal crosses. And I view that the Mwanga Diversity Panel is probably going to allow us to test those hypotheses in sweet potato. So we'll be looking at that also. My feeling is right now is first we need to make the tools, and we need to apply them to simple targets, and then build upon that knowledge.
There's no question, and please, if you have a question, you can raise your hand and then I'll see you and call you to ask a question. There's another question of how long has it taken to get the reference genome of a sweet potato up to this point?
Remarkably quick. And it's because of a unique confluence of beneficials, some great confluence of good circumstances. Probably the reference genome has developed in the space of 1.5 years, and the genome browser will be posted probably in the next 60 days. And then it's going to be an iterative process where we enhance that browser with improved resources and the community feeding back on that and annotating that are known through the browser. So version is going to be out pretty soon, and that'll be a first, and it's going to be an iterative process where we'll slowly improve it. But it's really come a lot quicker than I had expected, and I think it's because we had the right germplasm in place, we have new sequencing technologies, and we have a stellar team that's implementing it.
Okay. Questions from the audience?
I see their hand before. Ahmad, was it?
Muhammad Ali. You have your question?
Yes, I'm a master's student doing crop economics in China and basically from Pakistan. So I'm
Louder.
I
I also can Muhammad Ali.
Yes, I'm studying
Yeah.
in China. I'm a scholarship student. Then I'm asking that you are working in the North Carolina State University, and you are working with sweet potato, which is basically grown in Africa and some Asian countries like China. So how we can get benefits from assemblies and your research and other what you have done in the parliament of sweet potato. How the breeder can basically get benefits from the work you doing and not breeders mostly they are in Africa and they have no facility to do genomic research. So how we can get benefits from the work we have done.
Muhammad, we did not get your question clearly. Could you please use the chat and type your question?
Yeah, it's a good idea.
Most of the breeders are working in the less developed or underdeveloped countries. Can we get on this? Then I'm asking them how to get from the work they have done.
I've sent you a text. Kindly type your question because we can't hear you clearly.
Okay.
The next question.
He should close his mic.
Well, you have a question? Please close your microphone and your question will take it back. Ramjala, kindly ask your question. Your hand was raised up.
Go with your one more.
Yes.
Okay. Yeah. Ahmed will be texting or typing her question. And I don't know if there's anybody in the audience with more questions. Kindly raise your hand, and you'll have a chance to ask the question.
All right, okay. Yeah, I'm not seeing the.
Since no more questions from the great.
That's. Yep.
Okay. And so the audience will be, as I said when we were starting, this is the first of a series of webinars that will be the Genomic Tools Project will be organizing to educate and inform the breeders and the wider communities of practice who are on other crops with complicated or complex genetics. So as Craig put it, please stay tuned. We'll be sending invitation to the next webinar.
I'm sorry.
I have a question. Julia.
Okay.
Yes, please go ahead.
Okay. Thanks for the invitation. And my question is, or clarification, Tremendous progress has been done through conventional breeding, where there has been a lot of population improvement through different stresses. And I want to know what plans they have to characterize the local germplasm, and so with the reference to the mapping population, what do you know about the position of this creature in different backgrounds?
Good question. One, there's a lot of work done in many of the local breeding programs in sweet potato to characterize the local germplasm. But with the Genomic Tools Project, we're come up with a set of easy-to-use markers in the future and some protocols that can be implemented more rapidly in collaboration with, say, BECA or with the gene typing platform. So my vision would be is that we have a good pipeline. Maybe one or two of your students that might be working on that would do that, and we'd offer a service in terms of characterizing diversity. So I think that's an opportunity right there. In terms of the mapping populations, we're helping new mapping populations, but we have a very long way to go to actually use those. And so I think that for me, the analogy that would be was we're building a football field, if you will. And when we build that football field, we want more and more players to come play on it. But first, we need the stadium. So building the stadium, and then we have other people who can come and use that stadium, if you will. Does that answer your question? I mean, it's
Julie answered.
It's clear that this is just the foundation, and there are yet more things to be done on the characterization of the germplasm we have. Thank you.
Yeah, that's very true, Julius. Yeah.
Thank you for the question. It's very valid. I read her question, and our question, Craig, you can hear me. Her question was how can the breeders in the less developed countries get benefits from the work that the Genomic Tools Project is doing so far, and because as we breeders are mostly from less developed countries. Yeah. So how will the breeders from the less developed countries who are working on sweet potato benefit from this project?
You answer that question as our capacity development person, and I think you have a very good knowledge of that. You can signal that from our perspective.
Okay. So, can you hear me? Thanks, Craig. Yes, you can hear me. Okay, now this is how you benefit. Craig mentions that one of our objectives is to do capacity building, and this capacity building we are doing in Sub-Saharan Africa, and these are in countries which are less developed. And now what we've started doing is by getting people to come, because we are in collaboration with BECA, or the Biosciences Eastern and Central Africa, to get breeders from those less developed countries to bring them, train in BECA, and teach them how they can use the marker-assisted breeding to improve their sweet potato breeding program. For last year, a call went, and one of the avenues we are using is the African Sciences Challenge Fund, which is a fund which is funded by the Bill and Melinda Gates. And so we've gotten a few candidates to come here at BECA for about a month to 1 year to come and train on molecular breeding. The other avenues we are using is through workshops and through consultations. Like if you go to the Sweet Potato Knowledge Portal or to Genomic Tools Project website, we'll be posting a number of items that you can utilize and show in your breeding program.
I'd like to just kind of follow up on that a little bit too, is because I'm very sensitive to this, and I recognize the fact that most developed countries, particularly some developing countries in sub-Saharan Africa, really don't have access to these high-powered resources, and very much the way that I view it is sort of like you described. We'll have training and capacity development, but what I want to see and what we've seen through the SASHA project is a community of practices developing, and I would almost view them as overlapping circles in a Venn diagram. And so it might be that you could reach out to Becca, and you can send some of your technicians to Becca for training, or say Mercy can go there and give you some training. And they might be able to access NC State. So it would be that chain phenomenon, and we just build that capacity slowly. And I think it's important to know is my horizon is not the next 2 years. It's the next decade. We're talking breeding time. And my whole philosophy has been is